In this video, we will be discussing the process of telomere creation in eukaryotes. We will start with a short overview of DNA replication because telomere is the final step in new DNA synthesis.
This process involves many constituents. There's DNA polymerase, RNA primase, telomerase with an RNA template, and DNA ligase. Process of DNA replication, the DNA is split between leading and lagging strands. DNA polymerase is responsible for synthesizing the DNA along these strands, starting at the origin of replication, which is designated by an RNA primer, and moves in the 5' to 3' direction, reading 3' to 5'. For the primase to place the primer, a template strand must exist. This is an issue at the 3' end of DNA because DNA polymerase needs to replicate all of the existing DNA. The DNA on the 3' strand yet to be replicated is known as a 3' overhang, and this area is known as the telomere. Without telomerase, chromosomes would shorten. This would cause us to get old really fast.
Telomerase saves the day by lengthening the telomeres. A 3-nucleotide segment of RNA within telomerase causes it to bind to the 3' overhang. The protein subunit of telomerase, called telomerase reverse transcriptase, not shown, uses its own RNA template to add repeat sequences onto the telomeres. The RNA sequence beyond the binding site functions as a template for the synthesis of a nucleotide sequence at the end of the DNA strand. This process is known as polymerization. Because telomeres consist of a repetitious chain of nucleotide sequences, telomerase moves to the right and begins to make another repeat. This process is known as translocation. This process is repeated many times to lengthen the top strand. The telomerase then leaves after completing the set amount of repeats to make way for RNA primase. Once RNA primase enters and binds an RNA primer to the end of the newly lengthened telomere, the bottom strand can be made by DNA polymerase. DNA polymerase starts to synthesize the complementary half of the telomere. Once again, the length of the telomeres is important because the telomeres act as a protective cap for important genetic information towards the middle of the chromosome. Once the complementary bases are completely added, DNA ligase comes in and seals the bottom strand together. In order to complete telomere creation, another DNA polymerase must enter and remove the RNA primer. Although this still leaves a 3' overhang, the important DNA that is vital to life is now safely capped with telomeres.